Yes, good afternoon, everyone. Welcome to this afternoon session on decentralised manufacturing and point of care. My name's Brian Hanrahan from Planet Innovation, and it's my pleasure to welcome our speakers to the stage. So Gaudet from Galapagos, Marcos de Lima from Ohio State, and Lee Buckler from Advanced Therapies of Blood Centers of America. So if you can come up, gentlemen. As they're coming up, just a little bit of background. So for the session, it's interesting that Anthony Davies on Monday mentioned, that we're coming up on the 10th anniversary of the approval of autologous CAR T therapies, nearly 10 years with centralised manufacturing, and issues of accessibility and affordability are sort of the top of the tree in terms of challenges for the industry. Now, centralised manufacturing is going to continue to evolve. We have companies like Cellular Origins, Multiply Labs that are continuing to look at leveraging automation and robotics to meet or to address those challenges. But today we're going to look at alternative approaches with our esteemed panel. And so what we'll do is there'll be 3 presentations, about 15 minutes each. I'll get to hold the questions to the end, unless the guys are quick. And then we can have a bit more of a 10-minute Q&A to finish up. So to get started, Ian Gaudet is here from Galapagos. He's the head of US Regional Technical Operations and Vice President. His role is responsible for overseeing strategy, development and operations in the US regional manufacturing network in support of Galapagos' pioneering decentralised manufacturing approach. Ian has over 20 years of experience in the life science industry, including cell and gene therapy, biologics and small molecule discovery. Ian has extensive experience serving the cell and gene therapy market from within the contract manufacturing sector at multiple CGT organisations, including Chief Scientific Director at Monsanto Biotech, Director of Innovation and Engineering at Minaris, and Ian holds a doctorate in biomedical engineering from Rutgers University, where he has developed and patented a hydrogel biomaterial for use in regenerative medicine and tissue engineering. It's my pleasure to welcome Ian to kick it off.
Update everybody on, there we go, what we're doing at Galapagos, trying to improve patient access to CAR T therapies through our innovative decentralized manufacturing paradigm. Standard disclaimer, this presentation contains forward-looking statements. Please take everything I say with a grain of salt. As we all know, there are still significant limitations with current CAR T therapies in hemato-oncology. Ultimately, there's still a huge unmet need, and patients that need these therapies are not getting them. Particularly patients with fast progressing tumors, poor prognosis and shorter life expectancy could really benefit from improved CAR T therapy access, and we think can be accomplished through decentralized manufacturing nearer to the patients. Ultimately, there are a lot of patients that are eligible for these therapies, but still far less than half of the eligible patients do not get the therapies that they need due to limited capacity, both on apheresis and manufacturing, complex logistics, long turnaround times, ultimately restricted access, and we want to change that paradigm. So our vision at Galapagos is to bring cell therapies to the patients that need them to save lives. It's pretty simple. So we're taking a pioneering approach, doing things a little bit differently because clearly what we're doing is good as a field, but there needs to be improvement, and we're trying a new approach. Centralized CAR T therapy manufacturing has some limitations, as we've seen over the past 10 years. Cryopreserved cells include complex manufacturing and logistics. There are capacity constraints, the logistics of frozen products, and limited access to patients. And we're addressing this through a decentralized production model, which was recently validated both in the US and on an ongoing basis in European trials, leveraging our median 7-day vein-to-vein manufacturing cycle and delivering fresh, fit cells that have an early phenotype. These are manufactured in a very fast, rapid manner near patients, and we're showing that this is a globally scalable paradigm. So we aim to address limitations in CAR T access with our fresh cell therapy, as I mentioned, a median 7-day vein-to-vein time. So we have an innovative, decentralized manufacturing platform that leverages automation on both process analytics as well as information automation, enabling real-time monitoring of the cell journey from the clinic to the manufacturing site, back to the clinic, all within 7 days. We use our Cellular platform for our digitization efforts. Our fresh fit cells are manufactured in a decentral manner, and this provides simpler logistics, no cryopreservation, and ultimately a cheaper, faster, and we believe better product getting to the patients more quickly. And treatment is administered to patients with minimal wait time, and I'll get into the reason why that is incredibly important both for patients and for cost of goods. As I mentioned, our digital platform we call Cellular. This is an internally developed digital platform. We couple that with the Cocoon, as well as our proprietary innovative QC strategy. And this allows, again, that fresh product administration in 7 days, provides greater visibility to patients and physicians, and improves the patient experience. So our internally developed Cellular platform is an integrated digital portal that manages everything from enrollment to scheduling, apheresis, transport, manufacturing, production, testing, QC, batch release, infusion, as well as follow-up and data analysis. And this innovative, groundbreaking platform developed in-house over the last 5 years really enables the rapid turnaround time as well as visibility through the manufacturing cycle. It also facilitates rapid technology transfer through electronic batch records, no documentation needed, really accelerates getting new sites up and running, which is key to our model. It supports the end-to-end treatment workflow, and it supports continuous monitoring of the process and real-time review for the clinicians, as well as the cell processing team, kind of like watching your Uber and knowing exactly where it is every step of the way. Our manufacturing goes on in the Cocoon platform. This is a functionally closed platform. Most of you have seen it. If you have not, stop by the Lonza booth. They have a cassette there, and we'd be glad to talk to you more about the platform. It's an automated cell culture platform with single-use sterile cassettes. We couple that with our innovative and proprietary QC testing paradigm. And again, this yields fresh cells that do not need to be cryopreserved. So there's some more details on the Cocoon. Utilizes, as I mentioned, a single-use sterile cassette comprising a culture chamber. There's ports for getting things in and out in a functionally closed manner. And it has refrigerated media on board to allow for media exchange in a completely automated and closed manner, allowing end-to-end automation, functional closure, and process monitoring in a GMP compliant manner with a form factor that supports multiple units being installed in a very small footprint and facilitating cleaning and other GMP requirements. So really designed to be fit for purpose for what we're using it for. So our decentralized model really requires that we're making the same product at every site, which for anybody that has embarked on a comparability campaign knows is no small feat. The way we're able to do this is through what we call consistency by design. And it really encompasses making sure that every component of the manufacturing process and ecosystem is consistent from the outset. This includes standardization on manufacturing and QC equipment, documents and quality oversight, part of our model. Cellular ensures that the batch record is identical site to site. We have personnel training as part of our paradigm so that the training of new sites is identical site to site, and that is a critical component. And even the materials we provide so that all the materials are the exact same and there are no opportunities for small but sometimes critical differences to find their way into the manufacturing process that could yield a different product. And to support this all, we do have 24/7 support, frontline support for our manufacturing sites so that we can address any errors in real time and ensure that these products get out the door and get to patients on time in full. As I mentioned, we have a 7-day workflow. Starts with T-cell selection and activation, going into transduction, culture on the Cocoon, sampling, harvest, and formulation, It's pretty straightforward, very simple. And we do continuous production and QC review throughout the process to facilitate that very short window from apheresis to administration. And again, it's a 7-day process, vein to vein, which really is a game changer for patients that may not have more time than that, quite frankly. The proof is in the pudding. We're able to deliver this. This is some data that was presented late last year at American Society of Hematology by one of our clinical collaborators. And it shows that we are indeed able to deliver a product to patients as expected. In this cohort of, I think, 49 patients, 96% of them received a fresh product, 2 received a cryopreserved backup product because of clinical issues unrelated to the manufacturing process, but indeed patient health. And indeed, 43 of those patients did receive a product in 7 days, with 2 receiving a product in 8 days. So overwhelmingly, we were able to deliver as expected in 7 days to these patients. And importantly, that short vein-to-vein time eliminates the need for bridging therapy. So this is huge for patients to not have to undergo an additional round of treatment, and it's huge for the cost of goods, where that bridging therapy, hospitalization, is part of the holistic cost of these therapies. So we're not just addressing the cost of the drug, but indeed we're addressing the cost of the treatment, including taking out some of those components in the clinical setting that are very expensive, and ultimately making these more accessible to patients. Efficacy so far has been quite phenomenal. There was a lot of excitement when this data was presented at ASH. We'll go right to left on this data. Our mantle cell lymphoma cohort, obviously a small patient population to date, but 8 out of 8 patients achieved a complete response. In our follicular and marginal zone lymphoma, 95% complete response rate. And in our DLBCL cohort, which included a number of very sick patients with double and triple hit DLBCL, we achieved an overall response rate of 69%. And with our dose level 2, that went up to an 86% overall response rate and a 71% CRR. So the so far efficacy signal is very encouraging for our product. The slide's not going to tell you too much, but I always like to include it because a picture's worth 1,000 words. This is one of our patients with MZL, obviously a huge tumor burden. As you can see in the leftmost image, 28 days later, a complete molecular response, which has persisted to month 9 and currently are in an ongoing progression-free survival. And this is what we see by and large across the board with our patients. So we're seeing similar durable responses in all the cohorts that we have enrolled so far, with some ongoing responses going out to 2 years plus across all these responses. So it's not only achieving responses, but these are maintained, which is obviously an important part of these therapies being effective for patients and effective for payers. A subset of these patients did achieve minimal residual disease, negative status. And of those patients, there was 12 that were evaluable. All 12 of those patients are still in a complete remission and indeed progression-free survival 2 years out with some of the earlier dosed patients. So a very exciting signal on efficacy and durability. And we think that we have a good product largely because of the fresh nature, the short manufacturing cycle, and some other proprietary facets of our process that yields a stem-like early phenotype CAR-T product. And we can see across all of the products that we've manufactured so far, an increase in naive and stem cell memory products, a phenotype in our products, both in CD4 and CD8 cells. So we're not only maintaining that early phenotype, we're actually enriching that phenotype through that 7-day process. And this yields a product that is, we believe, safer, more efficacious, and able to elicit a durable response. And I don't have any slides on this, but the safety signal that we saw in our presented data was also quite encouraging. Out of almost 50 patients, we saw 1 grade 3 or worse CRS, 1 grade 3 or worse ICANS, both of which resolved with standard therapy. So a very encouraging safety profile that we think will be able to support outpatient administration, which again is going to reduce the cost of administration of these therapies. Some of the extra drug therapy costs can rival the cost of the drug itself. So if we're talking about a 400,000 CAR T product, these things ultimately can cost payers upwards of $600,000-$700,000. We need to get all of those costs down if we want to be able to provide access to these therapies. The cellular kinetics showed that regardless of the dose, we achieved a vector copy number that stabilized at about 100,000 copies per microgram of DNA, showing that this indeed is an expanding drug and that even at lower dose levels, we achieve the same level, albeit slower, of expansion in vivo. And we believe this also supports our hypothesis about the fitness of our cells, their ability to proliferate in vivo, and the ability to maintain a durable response. And indeed, seeing this out 21 months, in some patients, CAR T cells could still be detected. One exciting piece of news that was just announced yesterday, on the back of this exciting clinical data, we're emboldened to continue to build our manufacturing network here in the US with my team. Our newest site, we're proud to announce, is Catalent in their Princeton, New Jersey facility. This will serve a regional population of almost 38 million people. Within, you can see the image there, a 4-hour driving radius. So we have some pretty neat tools to look at patient location, clinical center location, and identify where best to locate our manufacturing sites using ArcGIS tools and some data analysis experts. So we think we can reach somewhere around 20 clinical centers from this site, ultimately covering almost 20% of the CAR T patient load just from one site. So we're very excited about this new site, and we'll be working to get this up and running to start enrolling patients as quickly as possible. So in conclusion, patient access gaps currently prevent many patients from receiving CAR T therapies that they need. A decentralized manufacturing of a fresh CAR T product addresses several of these gaps and allows for a 7-day vein-to-vein turnaround of a CAR T therapy. In our ongoing Atalanta-1 study, 100% of patients with refractory relapsed MCL, 95% of patients with R&R follicular lymphoma and marginal zone lymphoma, and 54% of patients with R&R DLBCL, again, 71% in our dose level 2, achieved a complete response. This asset, GLPG5101, demonstrates a manageable safety profile, again, with only one case of grade 3 CRS, one case of grade 3 ICANS in a reported cohort of 45 patients. And GLPG5101 is a fresh stem-like early memory phenotype CD19 CAR T therapy manufactured using a rapid decentralized platform, and it shows robust in vivo expansion and durable persistence that we think will be able to bring these kinds of results to more patients as we expand our network. So thank you for your attention. Be happy to answer any questions after the end of the session. You can go to Galapagos, or rather glpg.com, by using that QR code. Feel free to contact me after the show if you have any follow-up questions. Thank you for your attention.
Thanks, Ian. And that is some amazing data there. It's so exciting to see that, that is moving the needle in terms of patient access and treatment. So I'd now like to introduce our second speaker, and that is Dr. Marcus de Lima. Dr. de Lima is Professor of Medicine at Ohio State University and the Director of the Blood and Marrow Transplant and Cellular Therapy Programs at the Ohio State University Comprehensive Cancer Center. He has extensive experience developing and conducting clinical trials in the context of hematopoietic stem cell transplantation for myeloid malignancies. His research focuses on strategies to prevent leukemia relapse after transplant and the development of novel transplantation cell therapy approaches in treating blood cancers. Thank you, Dr. de Lima.
It is interesting that, Ash, we followed you. And again, here we are. So consider this a miniature of what he just presented. And there are several topics. So sub-themes here that I think are repetitive, so I'll try to avoid repeating those. So I actually have two hats. So I am here mostly as the CMO of a startup company founded by David Wald, who's sitting there, where when we started this at Case Western Reserve University, which was my previous job before I moved to OSU, just to see us win the football championship, if you haven't noticed that. I'm just going to leave it at that. So, and I'll base this, I was joking, but I'm not. This was also the concentration of my talk here is about data that we presented at ASH. So just follow, let me do a little bit of go back here. You know, this man, this picture here is Dr. Terasaki, who I think passed away like 3 years ago. So he was the first, he cannulated the thoracic duct and would get lymphocytes, right? This juice of lymphocytes, and was the first really evidence that lymphocytes doing adoptive immunotherapy had some responses, mind you, in cancer. This was when 1974. So cut. Now this explosion, right? I mean, we're living literally something short of a biologic, in my opinion, a biologic revolution, where we're still trying to make sense of this, as we all are, right? I mean, both from a market perspective, from a competition perspective, right? Nobody knows where this is going. But I don't know, you folks in business may know this curve better than I did. I didn't know that. But I think it applies perfectly to where we are now, right? I mean, we have this innovation trigger. You have this absolute inflated expectations. We're going to cure everything. And then we go zoom down. Now bispecifics are going to cure everything. CAR-Ts are not going to do anything. Oh, maybe not, maybe yes. So I hope, I don't know, you guys tell me, that we may be at that slope of enlightenment. You know, I want to believe that. Which would ultimately lead us to some sort of productive, you know, market or competition. So, but I don't know where exactly we are, but definitely there is also a little bit of a backlash, right? I mean, especially the competition with bispecifics, for example. So who knows, right, where that's going to go. So, non-Hodgkin's lymphoma, I don't think I have to elaborate too much on this, right? I mean, it's a disease that expresses CD19, and because of that, we have several products addressing that. But we believe, and that's where I think we want to convince you here a little bit, that we can make this manufacture very fast. And it will be about 20 hours. And for the same reasons we just discussed, right? I mean, turnaround time is measured in chemotherapy, right? Or disease progression, access. And I would say, you had a slide saying 20-30% of the patients don't have access. That's absolutely United States, right? I would extend to say there are whole continents that today don't have access to CAR-Ts, period. Right? Africa, most of South America, you know, half of Europe, it has erratic access, and huge areas of the United States, as you are trying to address, which is great. And then finally, those two things there, I think we can address also, as you mentioned, by changing manufacture. That is, maybe reduce side effects and maybe reduce relapse rates. So how do you do that? So potentially by having young cells, right? That increasing stamina. I don't want to, you already made this rationale. I don't think I have to repeat this. And I'll show you why we believe that may be the case. Now here I want to take a little longer. So think of the traditional process, right? Apheresis. You get mononuclear cells, right? You use T-cell isolate. You subsequently do your activation, right? And then expansion, and there you have your formulation. So what if you can start without apheresis? So you need blood. So you skip T-cell isolation. You definitely activate gene transfer, and you have your formulation. So by doing that, you absolutely shave costs dramatically. And potentially, again, back to the stamina hypothesis, right? So indeed, in David's lab, you can see one thing, that this very fast workflow, which is 20 hours. On the cartoon above is CD19 expression, below is BCMA. And on the right, you see basically traditional, like 7 days manufacture of cars. So you see, number one, the expression, both the CD19 and the BCMA is very similar. But if you go back to on the lower part of this here, you're going to see that a proliferation of the cells, you can see on the cartoon on the right, 1, 2, 3, the center, the comparison, how the cells proliferate much more, up to 100-fold more than the traditionally manufactured cars, being BCMA or CD19. And on the right, if you use this CCR7 marker for stemness, if you will, you can see also there, above, I think, CD4 and then below CD8, you can see the difference in this fast manufacture. So suggesting that this is happening, right? So what is the vector? So the vector has two novel domains. One is in the recognition domain, which is still towards CD19, and in a transmembrane domain that's proprietary. And if you look on the right of your screen, the lower is basically a mouse model showing how these cells again perform better than CD19 cars manufactured for 7, 8, or 9 days. Then we started and completed, by the way, a phase 1 study that involved the Cleveland Clinic in Cleveland, University Hospitals of Cleveland, which under Case Western University, and University of Iowa. So it's a 3-center small phase 1. Standard immunosuppression of diabetes cytoxin, and you can see there the description of the patients. Basically, a mixture. It's not just diffuse large cell lymphomas, but CD19 expressing lymphomas. Here, a picture. This is a patient that we look down there, had lymphoma in the bowel. So that's where that light is coming in the PET scan, and on the right, remission. The bottom line is, for the 12 patients that we have response, there is one more that we don't have the final response yet. We have seen an 85% response rate, complete remission rate. And the longest one now is 1.5 years. So again, preliminary data, but very strong preliminary data. The other thing that I want to highlight that goes with that hypothesis is that the toxicity rates seem to be very manageable. And there were 2 patients with a very, very low-grade inflammatory syndrome with CRS, and 1 patient that had 1 day of some neurologic toxicity so far. So nothing beyond that. So if you look, this is a comparison, right? I mean, from the data on the commercial products are essentially from the packaging cert, just comparing a little bit these rates in the clinic as they are now in commercial products versus our preliminary data in this initial cohort. Corroborating a little bit more this hypothesis is the cytokine data. So essentially what you see here on the left upper is baseline, and then day 6, and then down there is day 14, I believe. And essentially what you see is a modest elevation of IL-6, which is, goes with this low toxicity profile. And then finally, on this science side of things, is the kinetics of expansion reflect the very low cell dose that we use. So by virtue of being doing this harvest and infusing, we're infusing 17 million cells total. And essentially you see this expansion that instead of happening as usual on day 14, day 10 to 14, we're seeing that towards day 21. So again, goes with the hypothesis that you're seeing a more gentle expansion, and then because of that, you see less toxicity. I'm not going to elaborate too much on this, but there are so few non-responders, so it's hard to have a pattern in terms of, you can see in the Disney plot, one of the non-responders had the same profile as in the product as the person who achieved CR. So there's not a whole lot to say much here. So where are we as a company, right, at this point, as a small startup company? As I mentioned to you, we're expanding this phase 1 study. We have been working with centers outside the US as well. So we have a clinical trial agreement now with King Faisal in Riyadh to conduct a phase 2 study in the kingdom. And we're in conversations with 2 countries in South America to again replicate this and hopefully be able to completely decentralize, right? The whole goal here is to pass this manufacture, this process to centers to do it. We do have the next thing that's coming for us is a BCMA with the same platform, so to speak, which we are now, we got the IND, so it's the trial, David, is start now, right? I mean, I think we should be able to start accruing in a few months. I don't think we need to go through this, but I just want to highlight that there is still room, in our opinion, in the BCMA realm for fast manufacturing, although BCMA is also saturated, if you will, as a market in Europe and the United States for a variety of reasons. This is the preclinical evidence suggesting that this platform performs very well as well with a BCMA CAR. I'm not going to elaborate too much. And here, just to give you a hint that we are looking, we do have a target that seems to be doing very well against glioma and gastric cancer and a few other solid tumors. So this is something that we are hoping to be able to work on in the not so distant future. These are obviously our preclinical stuff. And here is where we are a little bit ahead. This is an animal model that, again, suggesting that this target, let's call it X, performs very well with this platform of manufacturing against gastric cancer in an animal model. So with this, I promise to finish. But I think the main message that I will say here, having developed 2 cell therapy programs myself, both in Cleveland and then subsequently Ohio State, and having helped a few countries outside the US. We did the first CAR T in Brazil and a few others. I've been around this business. From this academic perspective, I'll tell you one thing, that not everybody wants to hear this, but there is no other way to make CAR T more accessible than lowering the price. There is no other way. You may say, I'm going to do proliferation, I'm going to do the triple CAR, quadruple CAR. The only way to reach the places that are now not being treated with CAR Ts, or segments of the population, or the racial inequalities we're seeing, all these inequalities, the only way is to have a product that's cheaper. We can say whatever else, I'm going to give an assistance, I'm going to do this. It's not enough. Clearly not enough. We're seeing this in the United States. So we're trying to go that way. Our goal ultimately is obviously to commercialize this because it needs to, but we think that we can work seriously towards, reducing costs. But anyway, so that's it. Thank you very much.
Thanks, Marcos. I've been waiting for a couple of years to see someone to show some data where we did no expansion, and the simplification of that workflow is very impressive. So it's again, really interesting data. Our next speaker is Lee Buckler, I should say, and that's because he needs no introduction, I think. Lee is essentially, lost my little notes there. Lee is an experienced executive, investor, entrepreneur, and consultant in the cell therapy sector. He serves as Senior Vice President of Advanced Therapies for Blood Centers of America. He's a co-founder of Altruis Bioventures, a co-founder of the Advanced Therapies think tank, and is an advisory board member for several early-stage startups in the advanced therapy sector and industry publications. He's a founding member of Facilitate's Decentralized Manufacturing Working Group, one that you should all join. Lee.
Thank you, Brian. Is this on yet? Oh, there we go. Thank you, Brian. That's very generous, and I'm humbled to be on the stage with these 2 fellows. That's super exciting data that you're presenting. you've seen two very different models here of decentralized manufacturing. And the point is there isn't a single model that represents how to bring the manufacturing closer to the patients, closer to the clinical sites. But there's no doubt that it has to happen, right? One way or another, and probably multiple ways. I'm never a fan of binary. This is going to win, that's going to lose. I'm going to present to you what we're building. This is the least nice scientific talk you'll hear today. And so it should be relaxed on your brain and late in the day. So, the one thing we know for sure, despite the fact that there are different models, is that decentralized means not centralized, right? That we can agree on that. Whether it's in the hospital, whether it's in the blood center, whether it's a bedside machine, or whether we've completely eliminated manufacturing, we're certainly not doing it in big palaces of clean rooms, in centralized facilities, shipping LN2 tanks around the world, because for many autologous cell therapies in many jurisdictions, this is a rather unsustainable model, right? We think to drive patient accessibility. I have, by the way, you'll notice from the introduction of the speakers for this session, I'm neither Boro nor Fabian, but I did reach out to my friend Boro, who had to travel internationally because he's busy proselytizing decentralized manufacturing in other parts of the world. Borrow some of his slides, and so you'll see some of his slides here. We have, of course, lots of candidates of early stage ex vivo manufactured cell and stem cell gene therapies, and the beginning of certainly a transformative era. But as my previous speakers have alluded, it's certainly very high cost, and a cost that's not likely to be sustainable by healthcare systems in many parts of the world, if at all in the United States. And many predecessor speakers have spoken about this lack of meeting the eligible patients. I think it's not just a lack of meeting eligible patients. It's not that all of these patients are being prescribed the medicine and we're failing to manufacture them. In fact, they're just not being prescribed the medicine, right? So we have a lack of demand. The eligibility is not translating to demand. And I think we have to think about that very carefully, because clearly the prescribing physicians and the hospitals they're working in just aren't introducing their patients to CAR T-cell therapy, I think, in many instances. And then you compound that with a lack of access. Oh, he had another slide here I thought I really liked, but anyway, so be it. The percentage in lower middle income countries is abysmal, right? As Marcos pointed out. There's many solutions to addressing this. I think it's going to take all of this and a whole ecosystem of stakeholders. There's lowering the cost of equipment and materials. There's eliminating manufacturing, shortening manufacturing, tech transferring to hospital stakeholders or government systems in different countries, reducing the economic impact of logistics, all sorts of ways that all are going to contribute to better accessibility. No, there's no single panacea to this problem, but it's a problem that we all need to address in any number of ways. And like Marcos, Boro is busy transferring technology to other parts of the world, enabling in his model, and I'm here speaking on his behalf a little bit because he wasn't able to come to the session. And I think he and others like him are going to be an important part of changing the dynamic of cell therapies in particularly lower and middle class incomes countries of the world. In his words, able to deliver CAR T-cell therapy in a decentralized model and an all-inclusive cost of $35,000 per dose, which many people, to be perfectly honest, don't believe at all. But he says it many times, and I know he believes it, and I know he's delivering it, and has done a very exciting deal with the Brazilian public healthcare system, which is very invested in bringing this model to other parts of the country. And of course, we are, as Marcos alluded, at the very tipping point, but very early stages of what is certainly a revolution in healthcare based on this. We, following Ian's talk, as Blood Centers of America, are proud partners with Galapagos to support one of these visions, and that's in Galapagos' interpretation, the biopharmaceutical model of leveraging blood centers as proximate manufacturing environments near the point of care. So in our view, decentralized is de-linked from point of care manufacturing. Certainly there are hospitals capable of being manufacturing environments, but many of them aren't. Blood centers are accustomed to quality management systems and I&D and regulatory oversight and following protocols. And so last year, we announced a very exciting partnership with Galapagos where in the right jurisdictions where we decide that there's a matching footprint with clinical demand and we are the right footprint, we will provide a manufacturing environment for their point of care or for their decentralized manufacturing model. And we have in the room our first site, the CEO of our first site, which is Excelos in San Diego. Which is now in the middle of tech transfer, and we have other sites in progress and under evaluation. So I think what's interesting is the macro environment in which all of this is happening, right? We have this massive shortfall in CGTs meeting market demand. We have an increasing recognition of the geodiversity in biopharmaceutical models, right? And a general trend toward micro manufacturing, and also simultaneous with this, the evolution towards closing tools and technologies, automating tools and technologies, and digitizing every process unit. All of those are imperative to allow, if there's manufacturing at all of those are imperative to lowering the cost of goods, lowering the amount of time in clean rooms, if we need clean rooms at all in the future, and lowering the skilled labor required to do this manufacturing in bunny suits and clean rooms, right? There's this move toward, I think, what I like to call, the push and pull of any technology revolution, right? We have this market pull. Obviously, there's a huge unmet demand of patients who need these therapies. There's this technology push of all of these vendors out there innovating tools and technologies now built for the purpose, because this whole industry is built on tools that weren't built for the purpose. And we're moving away from paper forms and quality management systems and moving to digitization of every unit. And as you saw from Ian's talk, that model depends on be able to have real-time view of what's happening in every one of these centers. And you can only do that if you have a digital readout and you're able to have that kind of centralized oversight over your decentralized manufacturing sites, right? That's the great irony of all of this. And so we have, the push and pull, we have demand exceeding supply, and we have a regulatory pathway that is, I think, evolving and trying to keep up with the pace of innovation here, which is exponentially difficult for all of us, let alone for them. But we have, I think, a limited window to fix a problem here that is going to be a blight on our industry if we're not able to supply these highly innovative, highly curative treatments to the people that need them across socioeconomic barriers, across geographies, because there's too many stories of unmet need, and the last thing we want to be painted as is as a luxury medicine, right? No one gets up in the morning to say that's our mandate. And so we've talked a lot about cost. There's a lot of sort of innovation happening. This technology push I've talked about, it's not only around the closed systems and automation and digitization. We have a massive need in better analytics. Real-time analytics, obviously a 7-day, vein to vein requires advanced analytics where we're not waiting for 14 days for cultures to come back, right? This is unsustainable. We can't be releasing everything at risk. We really need to know what we're infusing. And so there's a lot of innovation happening here. And I think we're going to see next generation assays. And of course, AI and ML will help with the optimization of a lot of this. And so I think, the key pillars for any adoption like this is the technology readiness, the regulatory readiness, and the institutional readiness. Like Marcos's institutions and other hospitals, which are innovating around this as well. And, as much as this seemed even 24 months ago, like a science fiction, nobody believed decentralized manufacturing was ever going to happen. The regulators are on this, right? A year ago, they already were starting to do voice of customer industry sort of input gathering on decentralized manufacturing, certainly in the US, also the MHRA in the UK. They're trying to figure out how to enable the regulation of decentralized manufacturing units. wherever they sit, whether they sit in a CDMO, in a hospital or a blood center or some other manifestation that we haven't even predicted yet. So they are racing toward defining the regulatory pathways for this and setting up guidances and other sort of standard setting organizations are getting involved in how to make this real, because there's just too much at stake to put our head in the sand and pretend it's not happening in many parts of the world. And all of these are examples of documents and processes and programs that are being put out there to in fact enable all the levels of regulation and guidance that need to happen for this. And so, you know, there is a whole ecosystem of clinical environments. There's a whole ecosystem of tools providers, contract providers, digital solution providers, and AI ML providers who will contribute to this ecosystem that are innovating around this new paradigm of manufacturing. And there's just some samples of those on the clinical side, those some of the different frameworks of the model. And you know, this fits within, I think we're going to see in various countries, you know, not every solution fits the same in different countries. I mean, India is a prime example of some very, very rapid innovation happening, hospital-driven, because they just refuse to wait for pharma to come and bring their cell therapies, their CAR T cell therapies to India. And so they're innovating around regulatory models and hospital-based innovations. And I think we're even starting to hear murmurs of the regulatory agencies in Europe and America of starting to think whether there's a hybrid model for cell therapies in the context of the 351 pathway, right? We tried to fit cell therapy into this drug development pathway, and there was a lot of good reason for that. But I think there's some interesting push and perhaps even some exploration around whether there's a hybrid pathway for fitting drug development into expedited pathways, into hospital-based treatment modality pathways instead of the pharmaceutical drug development pathway that we are involved in now. I'll end with a little bit of futurescaping. I think my view is the future of cell therapy does not lie, for autologous cell therapy at least. Many cell therapies will not involve clean rooms, in bunny suits in clean rooms. I think we're looking at an evolution of manufacturing to, you know, 3D printing technologies, local print technologies, highly customized, personalized platforms where manufacturing is becoming miniaturized, localized, customized. That's a general trend that's happening across manufacturing, across all sorts of industries. And I think we'll be part of that evolution. And revolution, if you will. And I'm happy to represent the BCA footprint, of course, which, you know, represents 100+ years of community-based biomaterial supplies to hospitals in the United States, where we have a long history of donor recruitment, patient recruitment, biomaterial supply processing, governed by quality management systems, regulatory compliance in highly automated, closed systems in uncontrolled rooms for clinical infusion. That checks almost every box necessary for decentralized manufacturing footprint to hospitals, which are not typically manufacturing environments. And this is what the footprint looks like in the United States. This is our apheresis footprint, our cryopreservation footprint, our clean room footprint throughout the United States. It's the largest apheresis network. I think one of the largest, certainly the largest ISO 7 network of capabilities. And we've got a lot of work to do in terms of bringing this up to compliance and to standards. But it's a footprint we can leverage and a footprint we're excited to lend to advanced therapies as we work with different partners like Galapagos and others on bringing this supply chain ecosystem of, you know, 400 or 500 facilities across 70 members in 43 states with an enormous supply chain capacity of $1 billion worth of purchasing and $3 billion worth of revenue to bear for the supply of raw material, the supply of decentralized manufacturing footprint, and perhaps even exploration of infusion sites, outpatient infusion sites, as we move, up the supply to second line treatments, first line treatments, and perhaps outside of oncology to indications where the patients are stable, chronic but not acute, and perhaps don't need to be infused in hospitals. So we're very proud to be, leveraging this footprint, this experience of autologous and healthy donor apheresis, of therapeutic apheresis, and our footprint of 400 apheresis machines across 60 locations across the country to bear in support of this. And I'll just end with this as an indication of our ability to mobilize our network during COVID-19. We worked with the U.S. government to do 670,000 doses collected of convalescent plasma in a time which was hugely supply chain constrained, obviously. We were able to mobilize this network to supply convalescent plasma to BARDA to try and save lives of very acute patients with COVID. So happy to be lending our expertise and happy to be part of this ecosystem, and delighted to be working with people like this in the ecosystem. Thank you.
I think there's a microphone up there. So yeah, happy to open the floor to questions. Maybe I might just start with one. Ian, I was curious, and Lee mentioned, this challenge of QC and how do we do that. So I was just curious whether you could share a little bit on how Galapagos approaches, in-process control, that release testing, because again, fresh product says, how do we make that happen?
Yeah, certainly.
Stay there. You're right. I think I'll turn the mic on.
Yeah, we've invested heavily in our analytical suite, and that's been a big part of our investment in this platform, has been to make sure that we have methods that are both robust and simple, that can demonstrate comparability across sites, but also be rapidly executed to support that 7-day vein to vein. So it has taken a large investment, and we do partner with a number of analytical tools, developers, to leverage some of the groundbreaking technologies that can make these analytical tests as rapid as we need them to be able to release products, hours after they're harvested.
Thanks, Ian. Questions?
May I ask a question?
Yeah, go for it.
So how do you folks envision, say, if we truly decentralize, and then you are, I don't know, we mentioned Brazil, and you're here, your centralized facilities here. Do you think the technology is already here? So say a flow cassette that gives us the same gating every time, right? I mean, it's not prone to me changing the gate and saying, are things like that ready? And then the PCR that, I do it and then you do it. It's completely, it's a nightmare because anyways, what is your, right? Because I think the previous session he was discussing the variability in flow results, right? Right there, it's a classic example. The same market, the same almost people doing it, same machine, and then you get variations that can be one.
Yeah, it's a great point. There's been a lot of initial headway towards manufacturing platforms, but there was definitely a delay
the release
initially on the analytical tests. I think the good news is enough people have realized that this is a gap both for centralized and decentralized cell therapies, that we need more robust, more automated, more rapid methods. And so it has been an investment in the ecosystem. And I think we're getting pretty close. We will continue to augment our analytical suite with new technologies, and we do indeed plan to roll out several new methods that, like you mentioned, take the art out of these assays, replacing traditional flow with cassette-based flow, replacing qPCR with digital PCR, and really trying to take the human out of the equation, because that's always the most variable element in that equation is the human being. And so we're trying to minimize artfulness through leveraging some of the really exciting novel technologies that are out there. And I think we are there now.
Thank you.
Any questions from the floor?
Here we go. My main question is around commercial strategies. So you're proving a lot of ground with you just described here today. From Ian, from your perspective, are you also planning to supplement with some internal like pod or closer to location of your own company owned, but closer to the point care, similar to something surrounding some of these partnerships as business continuity? And then a little bit of a question for half the BCA element. When you move out, when you look at to globalize your program at a commercial level and the volumes jump up, and some facilities can take portion and you're assessing your volume, do you also see the need to maybe purchase some land, make an easable drop-in pod, or is any of that being considered? And for BCA is great here, and I don't know what your thoughts are on globalizing all that service or what other partners exist like you outside of the United States.
Yeah, that's a great point. Currently trying to leverage the large amount of capacity that exists with the CDMO space, but certainly there are areas where there isn't that infrastructure in place. And this is certainly where you saw on Lee's map, large coverage over the entire geographical region of the United States. Many of those regions don't have CDMO presence, and this is where we do expect to work more closely with BCA and partner with them to help invest and build infrastructure to be able to do decentralized manufacturing in areas where there isn't CDMO infrastructure. And I think that certainly translates to the rest of the world, where certainly we don't have that infrastructure in place right now. So we're using what's available right now, but certainly we expect to change that paradigm and work more with communities and blood centers and BCA and global stakeholders to really roll out access to these therapies on a global scale.
And from our perspective, it's a physical footprint of about 400 bricks and mortar across the country with a long-standing bio-supply chain relationship, within miles of all the regional hospitals without any necessary agnostic to hospital systems. So, and which own a lot of real estate. They own their facilities. There is oftentimes extra room available or greenfield space available in their locations. But a lot of them, let's admit, not everybody, there was 15 sites, ISO 7 sites across the network. About 6 of those are operating as a CDMO now. The rest of them are operating in some internal capacity or to support other things. And then the rest of them, don't have. But we are looking at, like germ-free prods or Hitachi systems that, with agreement of a customer like Galapagos on a pre-designed specification, we can fairly rapidly just plunk one in. They're standalone systems pulled into the parking lot or plunked into a garage, and get to work, fairly quickly, either, at the early stage or as additional capacity is required as a customer like Galapagos starts to ramp up. So we do hope to cookie cutter this across our footprint, which is admittedly just the Blood Centers of America. But what we're hoping is that, we can cut the die here and create a template that then we can help to spread around the world. It wouldn't probably be our footprint, because it's not within our mission, but we would certainly be happy to try and support, other countries, other institutions, other regions, to mimic the model that and the lessons where we've learned from success or failure.
Right now, you can see what regulatory concerns, especially in the United States, working with the CDMOs and working with bigger centers like Blood Bank of America, the bank networks. Would you envision that as the regulatory markers keep changing, that you really would be going down to point of care centers? Because you figure something like a Cocoon is not actually a really major investment at a hospital level to bring into a transplant center already, since they usually have some kind of stem cell lab or, under various names. Would you envision that really kind of coming down to that level, or would you want to see this sort of staying at larger centers where, it can be a little bit more controlled?
No, I think we need to figure out how to bring this manufacturing to community hospitals where it's going to be low throughput. And you're right, there isn't a huge investment needed to make that possible. And indeed, much of the data that I presented today was generated at point of care manufacturing in our European network. So it's absolutely feasible and probably necessary if we want to access some of the more remote regions where there just aren't going to be that kind of regional infrastructure in place.
And without speaking for Galapagos, I'll say, certainly my vision and maybe, is that this has to go beyond those hospitals with cell therapy labs. First of all, it has to go to community healthcare settings that don't have a stem cell transplant program, never heard of FACT accreditation, don't have even liquid nitrogen, right? We have to get to those places. And the more we can close, automated, simplify, push button, all of this, the easier that's going to be, because I think eventually it's not going to be even in an ISO 7. You look at blood centers, it's push button machines and dirty rooms, right? And we're safely transfusing blood and blood components every hour of every day.
Yeah, that's really fantastic, because that's really where it needs to go. I think even the transplant centers right now are trying to figure out how to really onboard those community hospitals and trying to help support them with the idea that, there is that expertise, and trying to push that now out to community offices. I guess my second question is, the numbers are absolutely fantastic in the manufacturing percentages. And I'm curious, for the apheresis process, so there is going to be variety in that apheresis, I would imagine, at the centers. How strict are you for making sure that products are really going to fit an immediate model to go into the manufacturing? Are there checks at that level that you might find that the apheresis really is not suited for that?
Yeah, you're 100% right. Garbage in, garbage out. Variability starts with the variability of the starting material. I think we have seen a push towards standardization on apheresis. I think there's standardization on technologies, practices, SOPs, but this is where I think working with BCA, where there already is standardization within their SOPs, is very helpful. And in addition, we are focusing a lot on ensuring that we have assessed what are the critical quality attributes of the apheresis, so that we can work with apheresis centers both within and outside of BCA to make sure that they are generating the same product that can then yield a comparable product once it gets into manufacturing. But it's a great point, and it's not a trivial challenge.
And on the flip side is the partner that would hope to us to supply those MNCA collections to a company like Galapagos. We're looking at ways to increase the characterization of what we collect. It's not enough to just collect per procedure and then not really know what we've shipped, right? I mean, we're looking to be able to better characterize and know exactly what we've shipped to Galapagos, and then we can try and optimize and control and at least trace, and then eventually correlate the variability of the starting material with maybe the variability of manufacturing and the variability of efficacy.
With the success, I would imagine that even as the transplant centers right now are really trying to push and educate community docs to get patients to collection much quicker, right? Before they're having all these devastating chemo treatments that are actually tanking their cell collections themselves. I think there's going to be a really good success as both sides sort of come at that.
Proactive apheresis makes a ton of sense. It's an insurance problem right now, but I think there are people really looking at that, at viewing apheresis at as soon as possible after diagnosis, in the event that it's required later on. And I think there's ways that we can create around that. And BC is certainly looking at that.
We're going to wrap it up there. But Marcos, you just want to make a...
I was just thinking that, or all these models and all these ideas, the next limiting step will be people. I mean, there is no, because here we're discussing 5% of the cancers, yes? I mean, my life looks like, everything is about leukemia and lymphoma, but truly, if you look statistically, these are 5%. So all it takes to break this whole conversation here is a successful CAR-T commercially approved for lung cancer, say. There is no way, I mean, maybe you, but we are already having trouble reconciling, for example, the wave of, which is not yet a huge wave, but of patients with melanoma. They're coming for tumor-infiltrating lymphocytes, or now synovial sarcoma, which has a TCR approved. And these are, tip of the iceberg. So, this isn't just a comment in general, but I think the next crisis, so I sincerely doubt that you're ever going to get to the community. I mean, at that level. I hope so, but I sincerely doubt, because it will be a bad capacity issue. There are so many other limiting steps that I can see right now, and I'm leaving this, that anyways, it's just, although I love it, I just don't see that happening at that level. I see it happen like that, closer to the community, but at the community, wow, that would be cool. But
I mean, unless you're really doing something in vivo, or you're doing like a Lupogen model where it's a box, you hook the patient up, you apheresis, something happens in the box, and you reinfuse in the same procedure, right?
yeah, sure. And then who is going to track 15 years of that patient from the FDA? I mean, the community are going to tell you, dude, I'm not touching this with a 7-foot pole because, but no, I love it. I'm with you. But I just don't think there's enough people for that.
Thanks, everyone, for joining us, and please join me in thanking the panel for a sensational discussion.